Well, and finally, I want to have a panel of researchers to talk about their experience working with diverse populations in research. And so starting from the far end and working toward me, we have Dr. Milap Nahata, who is professor of pharmacy and director of the Institute of Therapeutic Innovations and Outcomes, and has a special expertise in developing drug formulations for the safe use of children. And then we have Dr. Bradley Doherty, who is an assistant professor in the College of Optometry, where he studies the effect of stress and depression on vision, and so works with those special populations, also with, I think, an elderly population component as well. Dr. Mary Ellen Wiewers is a professor in the College of Public Health who does research in smoking cessation and works with Appalachian populations. And then finally, Dr. Lauren Sutherland is a clinical assistant professor in the Department of Emergency Medicine, and she has a special expertise in working not only with emergency populations, but also with elderly populations in that setting. So they're going to talk about their experiences in working with these populations, challenges in getting the consent process and retention, and ways they've met those challenges, and maybe some of the benefits of also this kind of research. So welcome to the panel. Thank you very much. I guess you guys can work among yourselves how you want to talk.
Would he like to go first?
Go ahead.
Oh. And I'm volunteered. Well, hello. Thank you for having me here. I'd love to talk to you all about what we do in the emergency department, because this is a really exciting place to recruit patients for studies when they have an acute medical condition. And the nice thing is we have a great research setup where we're currently recruiting for, I believe, 11 or 12 different funded studies, ranging from strokes, infectious disease, syncope, blood thinner use, flu study. Thank goodness flu season's wrapping up. So we have actually research recruiters who are constantly on the computer looking through everybody that walks into the ED and seeing if they might qualify for different studies. And we're currently staffed about 18 hours a day coverage plus half the weekends. So what we see, we go through a lot of patients. So it's nice because the patients come to us. It's difficult because they're often very sick, frustrated, in pain, and you have to be very thorough about whether you're consenting someone because they're willing to participate in a study or because they're scared they might have a stroke or scared of this disease and not take advantage of that. So what we found is that if you have a very simple intervention, so for example, our flu swab study, we're already sticking one swab up your nose, maybe we stick a second one and compare the two, a lot of people are comfortable with that. But the more intensive a study you're doing, people start to get worried that it's going to interfere with their care because everything's time-based. So you can't have a lengthy hour-long consent process because in an hour they could be out of the ER. But if you're looking to recruit patients during an acute condition or even to get them to follow up in your clinics afterwards, it's a great place. And though we have lots of trials and tribulations, we actually have a really successful crew and I'm able to complete a lot of good studies.
Well, thank you also for inviting me. It's nice. The panel, I think, brings very different perspectives and different groups of participants that we recruit. I work in the community, and Jill Oliveri is in the back row there, but we work in the Appalachian counties of Ohio. There's 32 of them. We have multiple studies, but my particular work is in the area of tobacco dependence treatment, and I work with recruiting smokers as well as some observational studies with non-smokers. So for the most part, our non-smokers are healthy, and our smokers are relatively healthy. They're people who, some people have a diagnosis, a tobacco attributable disease, but for the most part, I'm not seeing people when they're acutely ill or patient population. I can say that I actually just did the math in the back. We have recruited about 3,000 people from communities in Appalachia to be involved primarily in intervention studies where we could collect data longitudinally and try to follow people for a 12-month time frame. And I think as I talk more about our recruiters, you'll see why we have such good retention rates. We work with OSU Extension, which is an agency that's part of the university and has an office in every single county in Ohio. So it's already a built-in infrastructure that we can go to that county office, we can locate our field staff, have a home there. And the other thing is that in Appalachia, OSU, the university, has a lot of cachet, and to be associated and to be in a study that is connected with The Ohio State University is something that's attractive to a lot of people because we're thought of highly in the state. We also work in public health clinics, and we recruit, again, community samples, healthy people for the most part, but in some of our work, we need to have a nurse because we are giving medication, pharmacotherapy for tobacco dependence treatment. So even though it's over-the-counter medication, our IRBs like it if we have some healthcare provider supervision. So we often have nurses on our projects. And a couple of things I just wanted to say is we train our field staff. We have a reliable workforce that have been employed by us since the late 90s. And these are, for the most part, they're women. We've had some men, but they're mostly women, and we're able to hire them at 50%. We're able to offer them OSU salary, OSU benefits, and that's a tremendous advantage to us to have this trained workforce infrastructure in place. And as we continue to get grants, we are able to keep people employed and roll them over to new projects. And that's really important for the communities that we've been in for many, many years. It takes a long time to develop a sense of trust, and for people in the communities who we insist on hiring local community residents, it really goes a long way in terms of loyalty and trust and dedication to the research. We pay everybody. We pay extension agents. We pay clinic staff who don't work for the university, but we found that we get much better research, the integrity of the research and the dedication. Turnover is not an issue. They have to be county residents because they are, if I come from Columbus, I'm really an outsider, so we hire people from different counties to work in their own counties. And in terms of research retention, people in the counties know everybody, so they're able to find people. So for longitudinal studies, we have over 80–85% retention, and for tobacco research, that's pretty incredible because it's harder to retain smokers. So it really is an advantage to be able to hire people who live in the community where the research is being done. We pay all our participants for their time and their effort, so we give gift cards. We ask the community advisory boards to tell us, do you want a Speedway card? Do you want a Walmart card? Do you want a Marathon card? We get that information from the communities. And then finally, just the last thing I'll say is that over the years we have learned the importance of training interviewers to be respectful to the participants in the research. People go through Bridges Out of Poverty training, if you're familiar with that training, because a lot of time the characteristics of some of the people we recruit into our studies may be very different from some of the cultural characteristics of the interviewers. So it's important that people are comfortable working with all different types of people in research studies. So that's kind of what I do.
Well, thanks as well for having me. I'm happy to be here. As Stuart said, I work with people with macular degeneration. So these people are on average probably about 80 years old. They're dealing with active neovascular macular degeneration, which means that they have bleeding or fluid in the central portion of the retina, which is responsible for detailed vision. So they're going through a difficult time. They may be pondering the prospect of a lifetime of vision impairment or blindness. They are struggling to do some of the basic things that they're accustomed to doing, like cooking or dialing the phone or signing their names sometimes. And then on top of that, the treatment that's available for them, which is a recent advance and which is great, is an intraocular injection of an antivascular endothelial growth factor. So that does a great job of stopping the bleeding, but it's sometimes not a popular topic of conversation with the person who's already dealing with things. Well, the treatment is an injection into your eyeball. So that's disconcerting. And so for all of those reasons, and based on my clinical experience in trying to rehabilitate people with magnifiers and strategies after they've received treatment, we wanted to study stress and depression in these people. It's been shown that they deal with that at higher rates, and also the connection between those things and systemic inflammation and ultimately visual outcomes of the treatment. So the first challenge I think that we had is that it turns out that we needed people who were beginning a course of treatment right then and there. And in the College of Optometry, as I said, we tend to see them after they've already been treated for quite a while or they're done with treatment. And so that meant going to the retina specialist in the Department of Ophthalmology, which introduces a set of challenges. We haven't historically worked probably as well together as we would like. We operate different electronic health records. We have different buildings that aren't adjacent to each other. So there were a series of challenges. And step one was identifying a clinician who was seeing these patients. So we talked to the people we knew in ophthalmology and, had a few meetings and got to the right person. And from there, it was about, I think, developing a good relationship with that retina specialist, Fred Davidorf in ophthalmology, who's been fantastic, but that's been a multiple year process which still continues. So when Dr. Davidorf needs someone to fill in for him and give a presentation and he wants me to do it, I do it. And when they need help shuffling a patient from one room to the next or setting them up for some procedure, we do it. And I think over time we've been able to build a sense of trust that's been useful. And then we need to build trust with the patients too, because another challenge with working in a department that's not yours is that there are hundreds of patients here and none of them are mine and I don't know any of them. And that's a difficult thing when you go to obtain informed consent. So essentially we never approach the patient for the research without Dr. Davidorf. We go in together. He's known a lot of them for decades, and he gives a short introduction. He's busy. He's seeing maybe 3 dozen injections in the half morning or something. So he gives a quick introduction and overview and asks if the patient might be interested in hearing more. And I think that helps. Establish a sense of trust. And then we try to bring, I think, the philosophy that we have in vision rehabilitation to a different setting in ophthalmology. So sometimes we spend time answering questions about spectacles or magnifiers or just dealing with different disability cases, whatever, things that might not be the specialty of the retina specialist, but that we can talk about and again helps establish a sense of trust with the patient. So we've done that. We also try to pay people, so we pay for a portion of time of the ophthalmology technician, who is essentially the boss really there and is in charge of how the patient flows through the clinic and what the schedule looks like and who's coming up next and who can catch patients who are coming into the clinic who we want to try to recruit. So we make sure that that she's reimbursed for that time, and we spend a lot of time with the other technicians developing good relationships with them and trying to make sure that we're all on the same page about what we're trying to do. And I think trying to establish a sense that it's our research project and it's not that guy who always comes in here on Thursday mornings for some reason. And I think that's important. And then I hire people to cover all the bases. So someone has to be in the clinic just mostly waiting around until somebody who we want to recruit shows up. And I've seen people unsuccessful with sort of interdepartmental collaborations because they don't just put a body there to help the busy clinician. You know, Dr. Davidoff's fantastic, but he doesn't have time to, you know, check against my entrance criteria and perform informed consent in the midst of all the other things he does. So we wait there, and we just understand that a lot of the time we spend will be, you know, downtime, time that we work on something else, but we're there whenever a patient walks in and we want to recruit them. And that requires multiple people over multiple half days in different locations, and it's a challenge. The same goes for keeping track of the schedule. We work closely with their staff so that we always know exactly where everybody who we're looking for is going to be and when, and we can prepare for that. But it takes a lot of advance work, and we sort of learn that the hard way. And then ultimately, we're operating, as I said, in a busy clinic, so there's an element of not screwing up the people who are trying to provide the clinical care that the people came there in the first place for. So I try to hire people who know something about what's going to happen in this visit and what order things would probably happen and where the patient should be next. And we work, again, closely with the technicians, and we make sure that we're not hampering them in any way. All of our stuff combined, surveys, blood draw, visual acuity testing, you know, may take 30 or 40 minutes, and that's, you know, it's a significant amount of time, but we've pared it down to the minimum I think we can do and accomplish the research and provide minimal disruption to the clinical flow, and we work pretty hard on doing that, I would say. So they're mostly old-fashioned pieces of advice, I'm afraid, but they seem to have worked well for us, and I think we all probably have some of those things in common.
Well, good afternoon. Great to see all of you. I've been at Ohio State for only 38 years and still having fun. And I really feel very fortunate to be here. I came here to the College of Pharmacy. Didn't have a PharmD program at the time, so that was implemented the PharmD program, then established the clinical research program, first in internal medicine with gastroenterology, and then with pediatrics. Started with infectious disease within pediatrics. So my goal is to share with you some of my experiences. Bulk of the work that I've done has been in 3 areas. First is pharmacokinetics, the term that's used to describe how drugs are absorbed once we take them, how they're distributed in the body, how they're metabolized, and how they're eliminated. And assessment of those processes helps us determine what's the right amount to give to a baby who may be 2 days old versus a 2-month-old versus a 2-year-old or 18-year-old. And as the previous speaker mentioned, Grace mentioned that metabolism, absorption, distribution, elimination processes all vary at various ages. So that's one area, what we call pharmacokinetics and pharmacodynamics, to estimate the dose. The second is efficacy, safety, and drug-drug interaction type of studies. And the third has been drug delivery studies, because oftentimes the newborns have to be given drugs at very slow rates to make sure they get the appropriate amount and get a good peak, especially for drug like gentamicin or tobramycin, to get good peak above the MIC. And the drug delivery systems, when I began there in late 70s, were not very good. And the last area has been drug stability. A lot of drugs that we have are used off-label, so therefore they are available in tablets and capsules, and they can't be used in a 2-day-old or 2-month-old. So they have to be reformulated. But anytime you change a commercial dosage form, you have to look at the stability, palatability, how the drug is absorbed and handled by the children. So those are the 4 areas, and our research has been funded by pharmaceutical industries as well as NIH. There used to be what we call pediatric pharmacology research units. These were 4 centers that were funded throughout the US, and we were one of the first ones. So that work went on for about 10 years. And we were doing a lot of multi-center studies at that time. I've been also very fortunate to be part of IOM, the Institute of Medicine, and about 3 years ago they had appointed a committee for which I was a member to study how the FDA has performed over the last 10 years since the regulations changed for pediatric drug approval. Have they done their job? That was the question. So it was very important and useful experience to work with another 15 people throughout the U.S. to see how the drug approval process can be modified. So 4 things I want to emphasize today. Number 1, selecting the impactful project. What is the value of the project you're going to do? Is everybody excited about the project you're going to do? More interesting and more impactful the project, easier it would be to recruit and retain patients. And I'll give you an example. In late 70s when I started there, chloramphenicol was a drug of choice for treating bacterial meningitis. And we didn't have Hib vaccine, which we routinely use now. So at any given time, there were 5, 6, 8 patients with bacterial meningitis on the unit. And chloramphenicol was a drug of choice. And chloramphenicol used to be effective for the most part, but it has a lot of hematologic side effects. And most of them are dose-related. So it's not the ideal drug to use. So when a new drug called ceftriaxone, which Rocephin was a brand name, when it came out, we were among the first sites to use it. That has much better safety record. So to go to a parent to say, although the children are very sick, very ill, but to go to them to say that, look, we want to use this drug, which has better safety record. It seemed to work at least as good as chloramphenicol. It penetrates into cerebrospinal fluid much better than chloramphenicol. Our recruitment effort was extremely successful. Hardly anybody said no. Another example, otitis media, very common problem, ear infections. Every antibiotic had to be used for 10 days until a drug called Zithromax came out, and we were the first site. I was a primary PI site for the U.S., and that was the first drug that could be even for 5 days, once a day. It simplifies the regimen. So again, very easy to recruit patients. So my point is the value of the project, as the first speaker mentioned, what the value of the project, is everybody excited about it? Should it be, is it going to be impactful or not? If you do another Me Too, Cephalo, whatever, then it's not very exciting. There were a number of other drugs came out in the 80s, in the 2nd, 3rd generation cephalosporins. We didn't want to do those studies because they were not really adding that much value to what we already had. So that's my number 1. Number 2 is designing the study well. To make sure that you don't have too many restrictions in the study without compromising scientific validity and the quality of the project. That's very important. Inclusion-exclusion criteria should be right. Just yesterday I was talking with a friend in South Carolina who is doing a study in autism, and the inclusion criteria is the patient has to be 6 years and above and should not have received previously these drugs that they're studying, risperidone and aripiprazole. Those would exclude patients. And 6 and above is the age. Well, guess what? Patients have already received these drugs since they were 3 or 4. So he is having a tough time recruiting these patients. So somebody didn't pay attention to the protocol design. And the study is already going on at multiple centers. So this is an example of what was shared earlier, that 80% of the studies take too long and 20% never recruit very many patients. So really very important to get involved with the drug companies whoever the sponsor is, or it's an NIH-funded study, to get involved with other investigators to make sure that you design the project right. Number 3 is the ethical concerns. And Grace mentioned in detail. I would just only mention a couple of things. Number 1, the concept of normal volunteers, especially for phase 1 study when we're doing pharmacokinetics. All we're doing is giving the medication dose and collecting blood samples to look at how the drug is being handled by the body. If you were doing the study in adults, you could put an advertisement in the lantern that please, come and we give you a dose of the drug and we'll give you $500. You stay with us for 2 days and we get blood samples and it'll be no problem. But I don't think I can put an ad to bring your child in and we could give you $500 and you just can't do it. So normal volunteers are to be avoided. And we had a big discussion at the IOM about, is there a place where normal volunteer can be used in pediatric studies to improve recruitment? So one question came up, can you use patients that have history of migraine? They don't have migraine right now, but they have a history of migraine. Could you bring them in to study a new drug, although they're not experiencing headaches right now? So my point is, you want to avoid as much as possible the concept of normal people being given drugs. Number 2 is the payment. So we did give payment, but in most studies that I've done, we normally gave in the form of a bond in the child's name, usually $50 bond as a compensation for the study instead of giving to the parent. So there is no direct inducement. And the other point is the amount of money that you're giving. It should not be so tempting. $50 seemed very reasonable for 1 day study. If it's a longer study, it might have been $100 or $120. And the concept of placebo. That's my last point under ethical concerns. The placebo-controlled studies, if they must be done, you have to do them, but we want to avoid them. So, for example, in ear infections, when Zithromax came out, there's no reason to use a placebo-controlled study. Why don't you compare it to Augmentin, which is already a proven drug? So use a drug that's already on the market that works and compare that to the new drug instead of using placebos, especially when a child really can't give true consent. The 4th point I want to make is have the right team of people to work with you. Everybody must be excited. Especially my physician co-PI has to be equally excited as I am. The nurses have to be excited. The nurse coordinator, all the people, the team has to be excited about it. Otherwise, it just doesn't work very well. And you have to communicate with everybody all the time so that everybody's on the same page. And as was mentioned earlier today, that patients can tell, what's going on. So, and again, need to close the loop with them too, communicate very effectively with them, especially if it's a multi-day study, to make sure that they know what's going on, what's to be done when. And at times we've done hypertension studies, for example, where the patients were given ambulatory blood pressure monitor that were sent home to determine what would be appropriate safe dose for those patients. And they're attending schools or daycare. So the point is, who's going to give that extra dose if it is to be given during the day? So all those details have to be worked out, and having the right team really helps a great deal. That whole idea of caring must be so obvious. And the litmus test I always used was, would I enroll my own child? That's the test we have to use. And if we have concern about enrolling my own daughter, then I don't want to be involved with that study. So it can't be just to make money to run my other studies so that we can make money from here to do this study. That I never was able to convince myself to do. And finally, close the loop at the end when the studies are done, get the team together and share the excitement, celebrate a little bit, and publish the results. Present them and publish them. That's the message. So love to answer any questions that you have.
Yeah, so what questions we have this panel might have answered challenges you've been facing or been thinking about?
I've been involved in pediatric research once at Boston Children's Hospital, but I'm intrigued with the information that was passed on earlier about the ascent age. Some institutions 7 and 9. Can you tell me a little bit about the history of that, and what does the NIH define it? Are they particular with that, or?
Well, if you look at our report of the IOM, we avoided specifically mentioning a specific age. We let the institution determine that because there was a controversy. Some of us felt it should be as early as 4 or 5. If the child says no, it's a no, period. How can you argue with that? So we didn't want to put a 7 or 9 because I think the child's participation is very important. At least that they're interested in, at least they understand they want to go through it. That is very important. So we did not put any specific age there. So they're not dictating any specific age. Most places, as was said earlier, is 7 or 8 in majority of the places. But I think many places feel that it should be lower than that. But then you don't need signature though. Under that scenario, just a verbal, okay.
Other questions?
You know, most of you, not necessarily in the ER, talked about trust and caring. And so those are the key things. I find it's much easier to recruit my own patients than it is somebody I don't know, because I've already established a relationship of trust. And so a large problem is when an investigator who's not the patient's provider, like in your situation, wants to use those patients, and how do one convince the I mean, what you said makes the most sense. You have to have the provider, the person they trust, introduce you. You have to support their staff for spending all the time with you. They have to provide you space, and so somehow you have to compensate them. And so those are immense, expensive hurdles.
I've noticed.
Yeah. And so I guess that's the issue. Is there ways to short circuit this?
Yeah, I don't know. I sort of doubt it. We've been lucky and a little creative and been able to like I said, to do just those things. And it actually, interestingly, it hasn't taken long. You know, we say these people are repeat customers at the retina specialist. So now when we get there in the morning and we walk into the lobby, I mean, we're saying hi to the people too. And so in some sense, we've almost recruited all of the patients that this particular retina specialist has that meet our criteria, and they have, you know, not in the same way, but in some sense become like our patients too, and that's helped. It helps we follow them over a year. So the first one is difficult, and the first one requires the most care and the most interaction with the clinician that they're familiar with. And after that, it becomes easier on follow-up visits. But in terms of short-circuiting the process, you know, I don't know. I don't have an answer for that. And in fact, it's stopped us probably from going to other offices in an effort to recruit more people, because I don't know a shorter way to do it, and it's been an immense project just to do it in one place, and a long-term project.
So Jeremy showed a slide up there of something about where the MyChart message comes that you qualify for somebody you don't know's study. Yeah. If that message also said, and your Dr. X, who cares for you, says you might be interested in this, do you think that's enough of an entree? I mean, being referred I mean, that's what a lot of situations is. These aren't your patients. You're depending upon somebody else to do the introductions. They're not there. They don't have the time to be there. I mean, I don't even have the time to talk about my own studies with my own patients sometimes. And so I wonder if these other ways to notify them in the mail, on the MyChart message, would those work? To let them know that, hey, here's a study, I think you might be interested.
Yeah. It seems like
Anybody other
yeah.
ideas on that?
it's a step in the right direction, although my gut says it's probably not enough.
Not enough.
And for our patients, you know, they're 85 or 90, maybe they can't see either. So a
Read
lot of
the
these
email.
things, I hear about these great new inventions for recruiting, and I go, boy, I wish I could use that thing. But I don't know. My gut is that people wouldn't see it necessarily every time. Some might as enough, though.
I'm always amazed when my research recruiters can get anybody to sent to a study, because they literally just walk into a room and say, "Hi, I'm Danielle. I'm here at OSU, and I'd like to talk to you about a possible research study you may qualify for." And some people do just say, "I'm tired. I can't take it." But a lot of people really understand, and I think you made a good point about the name of OSU. They expect us to be doing research here, and they're not surprised to hear about studies. And some people are flattered to be asked to join a study. But we will often have the research recruiters call the resident working on the case and say, would this be a good person or not? And sometimes they'll say, no, she has schizophrenia, don't go in the room. Or they're really upset at not having sandwiches in the ED today, don't go in the room. So there are a lot of little things, and having the worst possible scenario to come and talk to somebody about a study, and we still manage to get pretty good recruitment rates and have a good NIH trials that we are successfully recruiting for. I don't know if you know about the PROACT study, the procalcitonin study we're doing. We're the second largest recruiting site on that right now. So it has to do a lot with the friendliness of your staff and the situation, but I can see on the outpatient setting where you don't have that pressure of acuity of condition to help you with your consent process, how difficult that would be to do the cold call. But if you want
training to your recruiters,
they have training. We have kind of scripts they go through when they talk to people, and Rose is trying to help us do even better.
Well, Grace mentioned how the grandmother helped in some patients. So maybe the certain kind, the person recruiting maybe makes a difference too.
Oh yeah, we had one of my recruiters came back and said, I talked to this lady and she was so nice, but she was 93 and she said, a blood sample. I just don't think I have much more blood to give. She said, that's okay, ma'am. It's all right. You have a good day. We'll go talk to somebody else.
I don't know, again, that being in the community and actually going for population-based samples rather than convenient samples of patients, but something we've done recently that's worked is what's called address-based sampling, where we have randomly selected household addresses in communities, sent out 2,000 letters explaining the study, putting a $2 bill, the research, there's been a lot of research on this, putting a $2 bill, not a $1, but a $2 bill, and then asking the household to enumerate the members of the household. One study we were looking for women 18 and above, and I thought, well, this is kind of creepy. You know, households might not want strangers to know who lives in the household and how to contact them. But we got a 44% response rate, and which out of 2,000, that's a pretty decent number for depending on your study. And then when we contacted the households that did send the questionnaire back, we got about a 75% participation rate. So, I mean, I don't know if you have that opportunity in patient populations, if you're allowed to send letters to your patients, but if there is a lot of research on this methodology, and the $2 bill being kind of the catch, that people feel obliged to at least fill out the questionnaire and then let you call them and give them the talk about how it's important to be in this study. So
Your $2 bill is sitting on my desk. We got one of the studies.
yeah,
I haven't filled it out yet.
We've just sent out 180,000 $2 bills for a Franklin County and a Appalachian. Yeah, so the federal government paid us a lot of money for that.
They print
I'm sorry?
They print the money too.
No, it's funny, we had to hire security here at campus to find a place to store the $2 bills. So.
Well, so I'm helping recruit. I'm a study coordinator, and I'm helping recruit for a study. And we are working with breast cancer patients. And so we actually have access to IHIS and also use Information Warehouse. And so we're allowed to screen doctors' schedules that we've talked to and that have a good relationship with the PI. And we actually have a breast cancer survivor that's a volunteer for our study, and she volunteers and goes in and talks to the patients. So she's kind of their first point of contact, and that's worked really well for us. So we'll call the doctors, say, hey, there's so-and-so, they're going to be coming in next week. Do you mind if, this woman comes in who's a breast cancer survivor and comes in and talks with them after they're done? And so it's just kind of their first point of contact. She sees if they're interested. If they are, she gives them more information, and then they call us. And that's worked really well for us. But we have a very specific population, and we are lucky that we have somebody who wants to volunteer and help us out with that. But I don't know. I think having somebody who can relate to them on that level is a little bit better than just me coming in and being like, hey, do you want to participate? So anyways, that's just my tidbit. So.
Any other questions or comments? Well, thank you all for your attention today, and thanks to our final panel.